Good evening, everyone. My name is Mansi Tripathi. I am TA for this particular course, that is Applied Environmental Microbiology. So, so this is the first week of the session. I am a PMRF student and a research scholar at IIT Madras. So, I'll be taking this doubt solving session. So, before sharing my screen, I would like to say, in case of we'll be solving few questions, and in between questions we'll have some proper discussion. So I want it to be interactive. You can reply to the questions in the chat box also, and you can say also, no problem. Also, what we will do, in between there will be a few parts of discussion which I find important. So apart from that, if there is anything that you would like to discuss in the next meeting or something, you can just let me know. You can type in the chat box also. So I'll share my website as well, so that if in between the week you have any doubt or any questions, you can just send me a message there itself, and I will be taking that question in the next session. So I'll share my screen. Just let me know if it is visible. Yeah. Is it visible?
Yes, ma'am.
Okay.
Ma'am.
Yeah. Thank you so much. Yeah. So, as I was saying, this course is Applied Environmental Microbiology. So, I am not aware of your backgrounds. That are you well aware of this course, or you are from PG background or UG background, and what kind of backgrounds do you have? So, I am basically a civil engineer. I have done my BTech, then MTech in environmental engineering. After that, I am doing PhD in environmental engineering. So, environmental engineering is the past part of civil engineering, which deals with water, wastewater treatment, and all those things. So, why, first of all, I would like to say why I am interested in this course, because when I am teaching something, I am solving doubt in something. First of all, I should say why it is important for me. So, most of you might not be aware, when we say environmental engineering, it says water and wastewater treatment. So, if you guys have ever been to water or wastewater treatment plants, so what happens specifically when we talk about wastewater treatment plant? For treating any kind of water, any kind of wastewater, we use microorganisms to treat our water. Okay, so that is where this concept is important, that how we are using the microorganisms to treat our wastewater. Apart from that, we know that their microorganisms causes diseases. So for removing these microorganisms from the system so that they won't cause us disease, we use various disinfection techniques. We use chlorine tablets, or just by simple keeping in sunlight, and all those things. Okay, so this is also the part of environmental engineering. So that is why I am interested in this course. I want to know about the microbes so that I can treat my water and wastewater in a better way. Okay, so keeping that in mind, that's why I am taking this course. So like that, you all are might be from different backgrounds, and all of you might be having different expectations from this course. So in due course of time, I'll be going in the basic level, and also in this course I won't be taking any lecture. There won't be any seminar kind of thing. This will be mostly, I will show you some sample problems that will be quite similar to the problems that you may get in the assignments as well as in the examination. But I would like to help if there is any doubt you people have. Apart from this, the Gargi ma'am has already given you lectures and everything. So all those are very exhaustive. So if you read it properly, if you go through them properly, okay, pause, play again, rewind, make proper notes, then this concept will be very clear to you. So for me, for my PhD, her lectures were very important. Everything is there that I can take some important information out of it and can use that in my technique. So like that, I would recommend to go through it properly. So this is for the first week. So this is the course content for this week. We have seen some history of environmental microbiology. After that, we have seen the microscopy, different kinds of microscopies. Apart from that, we have seen the cell, what is the structure, composition, different kinds of cell. Then is cytoplasmic membrane and transport. Further, we have seen what is DNA, what is gene, and what are the genetic elements. After that, there was an interesting topic that was central dogma. So here we studied about replication, transcription, and translation. These three were very important points of central dogma and the part of environmental microbiology. So this was the course content. So before jumping into the proper MCQ based questions, my first question is, like, we have studied microscopy, right, after the history. So history is also important if we want to know how a concept or how his knowledge is flowing in the time. To know that is also very important, but that is the part of history. Second point was microscopy. So my question is, why microscopy is important in microbiology. So when we talk about microbiology, what do these microscopes do? Why do we need them? Is there anyone who can answer? You can unmute or answer, or you can write in the chat box. So it's very simple. Like, you might have seen microscopes in your earlier childhood in school days also. So I would just like to know, how is microscopy important in microbiology? So is there anyone who can answer? Yes, very nice. To visualize microbes, nice. To identify the structure and components of the microbes, yes. So the important point is, like, this comes out of history also, yes. Most important is, it is invisible to the naked eyes. Yes, very nice. So when we talk about history, so when the man came to to some senses, there were diseases and all those things. So nobody was aware that these microbes can cause diseases, because no one can see that. So slowly, when microscope was developed, people came to know, okay, there are organisms or there are particles which are not visible to our naked eyes, but they are causing problem to us. So to visualize, to see all those invisible items, microscopy was used. So that was just my a general question. Okay, so the first question is, halophile microbes are mainly found in contaminated river with acid mine drainage. So is it true or false? I hope you remember different type of microbes: thermophile, mesophile, halophile. Okay, I am getting so many false here. So let's see the answer. So the answer is true. Okay. So what are halophiles? Halophiles are how thermophile is related to temperature. Halophile is related to what? Is there anyone who can answer? No, not light. Yes, correct.
Salt.
Salt, yes, very true. So halophiles are related to salt. So when there is water contaminated with acid mine drainage, there is an increased amount of salt in the water body. So that is why halophile microbes, phile means loving. Okay. So the microbes that like to live in high salinity, they can live in acid mine drainage water. So that is why the answer is true. Okay. Coming back to next question. Human microbiome affect human health by all the following except Please answer this question. I think one of the option is correct. Human microbiome means the microorganisms that live inside human body. They affect human health by which of the following? The health can be negative or health can be positive. The effect can be negative also. Effect can be positive. Think more. Think. Read the question properly. There is one option which is wrong. Okay. Human microbiome affect human health by all except one. So what is that thing that human microbiome does not affect? Okay, so you have to mark which one is not affected by human microbiome. Okay, so I have many responses here. So I'll see the answer now. So the answer is altering bone density. So supplying vitamins, I will give you example. There are few microbiomes which gets pathogens that when they get into our body, they start taking up vitamins. Okay, so vitamin absorption by the human body is affected. How elevating depression? See, if the there is one concept, brain gut issues, brain gut symbiosis. So you know that there are various microorganisms which are present in our stomach for digestion of food. Okay, that is right. So what happens when the digestion is not proper? You will not feel good, and when you won't feel good, there will be problems that you won't be able to do things properly. Okay, in your daily life you will feel there will be problems like while driving or something. So that is why sometimes the brain health is very directly related to gut health. So if the bacteria in our gut, the probiotic bacteria in our gut, if they are good, okay, then it will be it is kind of not directly but somewhat related to brain health, that is depression. And immunity, of course, if there are pathogens in our body, our immunity will be compromised. Fourth is altering bone density. The bacteria, they cannot do anything to bone density. So what alters bone density? There is one mineral that alters bone density, that is calcium. So bone density is related to calcium. Other things can be one way or another related to human health related to human microbiome. Okay. So the next question is endospores and the method to prevent contamination in the laboratory were discovered by. So this question is from the history. So who was the one who discovered first endospores, and secondly the method to prevent contamination in the laboratory? There will be space between the and laboratory. Please take some time to answer this question. I have one response. Yes, okay. So the answer is correct. This is kind of memory based question. Here you have to jot down your memory. So the next question is use of the world's tiniest pH electrode is not a characteristic of Sanger sequencing. Sanger sequencing is one thing. Read the question properly. The thing is it's not a characteristics. Okay. Accordingly, you have to answer true or false. What is pH electrode? It is used for measuring the pH. pH is what? Acid. It is negative log of hydronium ion concentration, negative log of H plus ion concentration. Is not a characteristic. Okay, read the question very carefully. Okay, we'll see the solution now. So it's true, correct. Okay, it is not a characteristic of Sanger sequencing. This question is very simple, bit tricky, So environmental microbes play important role in disinfection. True or false? Do they play role in disinfection process? Take some time to think. Are they part of any disinfection process? Are they doing any disinfection? When we say disinfection, we talk about all those things which can disinfect something. So, do we apply microbes to disinfect something? No, right? So, the answer is false. Good. Okay, I hope you understood. Here, they do not, they are the, actually microbes are the organisms on which disinfection is done. Why we are doing disinfection? Because microbes are there. If microbes are not there, we will not do any disinfection. Okay, so the process of disinfection itself is existing because microbes are there. So if we say disinfection, it means we are talking about all those chemicals, UV light, ozone, all those things which we are using to remove these microbes. So the next question is, when considering the primary difference between archaeal and bacterial cell membranes, one of the following statement is not true. Okay, not true. We are talking about prokaryotes, okay, both of them, archaea and bacteria. Cell membranes, that is what we are talking about. Bacteria have ester linkage with glycerol and archaea have ether bonds. Archaeal membranes are monolayered unlike bacterial membranes. Unlike archaeal membranes, bacterial membranes never have rings and sugars. Given structural difference between archaeal and bacterial membranes, it can be safely assumed that one of them is structurally fit for survival. Please take some time and answer this question. Okay, since I have two responses, I would like to show the answer. So the answer is correct. Option is D. Given structural differences between archaeal and bacterial membranes, it can be safely assumed that only one of them is structurally fit for survival. So that is obviously not wrong. We know that the archaeas are also there, and obviously archaea can live in highly extreme environments, and bacteria are also there. So both of them are structurally fit. It's not like one of them is fit. Okay. So it takes us to our next question. Which of the following transportation pathway is best suited for transporting a compound through the cytoplasmic membrane against the concentration gradient? Diffusion, simple transport using PMF, group translocation, or ATP transporters? Cytoplasm, here we are talking about cytoplasmic membrane against concentration gradient. Okay, we are going against concentration gradient. And what is the process um, which involves uh, this concentration gradient? The transport of fluid from one membrane to another with concentration gradient. What is that procedure called? You might have heard about this term, osmosis. There will be a semi-permeable membrane, and either side of that there will be, yes, osmosis. There will be, on either side of that there will be one low concentration material and other side high concentration material, and there will be transportation. That process is called osmosis. Okay, and when we use this process in the water treatment, we have used reverse osmosis, RO, in our homes, right? So that process is just the opposite of osmosis, called reverse osmosis. So that is how we get clean, pure water at our homes and other treatment plants. Okay, by the way, coming back to this question, let me show you the answer. So the answer is ATP transporter. Okay, diffusion process is like osmosis. It works in the side of concentration gradient, not against. Okay, so ATP transporter is the process which work against, and it involves cytoplasmic membrane also. Not all process involves cytoplasmic membrane. Ma'am, I have a question. Do we consider all the microbes like bacteria, fungi, microbes? What is the difference between required useful? Yes, first of all, we consider all these bacteria, fungi, virus, even algae. Okay, the green color particle that we see over the water body, that is algae. All of them are microbes. Microbes means micro, which having their diameter in terms of micrometer, that is 10 raised to power minus 6 meters. So all of the particles that are in that range or below that range are called microbes. And of course there is difference between, see, first of all, none of the microbes are useless. If they are not useful for us, they may be useful for someone else. Okay, but there are obviously good microbes and bad microbes. Okay, so if those microbes are helping in food digestion, they are good microbes and they are useful for us. If they are coming into our body and causing some disease like tuberculosis, that microbe is bad for us. Okay, but that is not useless, that is good for someone else, that is good for at least that microbe itself, है ना? So first of all these things are microbes only. So coming to next question, the bond between the pentose sugar and the nucleoside in DNA is glycosidic bond. Is it true or false? The bond between pentose sugar and nucleoside in DNA is glycosidic bond. Is the name of this bond is glycosidic bond? Answer in true or false? So the answer is true. The bond between pentose sugar and nucleoside in DNA is called glycosidic bond only. So the answer is true. So going to next question. The boundary between external and internal environments is not clearly defined and both have influenced on each other. Okay, is it true or false? Take couple of minutes to think. So the answer is false. Okay. The boundary between external and internal environment is very clearly defined. Okay. And obviously both have influence on each other. That is there, but this not thing is not correct here. So that is why it is false. And in any place, when we were talking about osmosis also and the other process also, what we have seen, there is always a membrane or the boundary, and that boundary is clearly defined. Okay, that this is internal and this is external. That is why this solution is false. The next question is, Pasteur tested the theory of spontaneous regeneration by. So this is from the history lesson. Allowing freshly cooked food to rot in laboratory, eliminating inflow of air in media and noticing no growth in it, by observing the microbes spontaneously exist everywhere, or by discovering the technique of pasteurization wherein a food item is heated under high pressure to kill all microbes. What can be the solution for this? See, the important point is how he tested the theory of regeneration, spontaneous theory of regeneration. Obviously, D option is correct. But is this correct? Is this D option correct in this question's context? Hmm? Now I have the enough response, so I'll show the solution. So eliminating inflow and air in a microbe and noticing no growth in it. So that one is the wrong option. Pasteur theory, it is the correct option here. And the one person has said option D. That is also correct, but that is not correct in this question's context. Okay. We'll move to next question. So identify the true statement regarding eukaryotes. It lacks membrane bound organelles, lack membrane bound nucleus, DNA is bound with histones, or none of these. We have learned about prokaryotes, eukaryote cell, right? So which of the option is correct? Please take some time to think and let me know the answer. solution is C. DNA is bound with histones. Okay. And we know that eukaryotes has very defined membranes. Okay, so option A is, and B is definitely not correct here. So C is the correct option. The next question is, which of the following are not a prokaryote? Bacteria, fungi, gram-negative bacteria, or protozoa? Which of the following is not prokaryote? More than one option is correct. That is the hint. So the answer is B and D, fungi and protozoa, because all of them has proper membrane. So the next question is, dash has its own genetic material that is distinct from the genetic material inside the nucleus of a eukaryotic cell. Ribosome, Golgi apparatus, endoplasmic reticulum, or mitochondria? Take some time. Shall we see the answer? So the answer is D, mitochondria. Mitochondria is the only thing that has its own genetic material. Okay, so in the nucleus there will be genetic material, but mitochondria will also have its own genetic material. And mitochondria, we have another name of mitochondria. Who can say? You can write in the chat box. We have another name of mitochondria. A very common name of mitochondria. Mitochondria is very scientific. There is another name. That is powerhouse of cell. Yes, correct. And why do we call it powerhouse of cell? Correct. It generates ATP. That is adenosine triphosphate. So we'll move towards next question. So this is the slide that I have prepared for you to understand the difference between prokaryotes and eukaryotes. So first of all, first is the diameter of cell. We can see that the size of the eukaryote cell is more than the diameter of the prokaryotes. So as I was saying, why we call them microorganisms? Their sizes are in micrometer. We can see, right, micrometers, 0.2 to 2. For eukaryotes, it's 10 to 100 micrometers. For prokaryotes, when we talk about nucleus, there is no nuclear membrane. And for eukaryotes, there is a true nucleus with nuclear membrane and nucleoli. Okay. When we talk about membrane and closed organelles, this thing is absent in prokaryotes, but eukaryotes, it is there. And we can see is about flagella, cell wall, and other plasma membrane. Okay. So what about cell wall? Cell wall is mostly pertaining to plants. Okay. So algae is a kind of plant which is which is also microorganisms. So when we talk about Prokaryotes, this cell wall is usually present and it is chemically complex. And eukaryotes, it is present but chemically simple. It is not always present. Plasma membrane in prokaryotes, no carbohydrates and lack strilols. Eukaryotes, strilols and carbohydrates are present. Cytoplasm in prokaryotes, no cytoplasm, and eukaryotes, there is cytoplasm. Ribosomes are smaller in size for prokaryotes and larger for eukaryotes. Same goes for DNA arrangement, that there are no histones. And what are histones? Is there anyone who can answer in the chat box? And for DNA arrangement for eukaryotes, there are chromosomes with histones. Yes, they are proteins, but what is their function? We are talking about DNA arrangement, how DNA is arranged, and what is the use of histones there. Actually, these histones keep the DNA arrangement in a proper membrane. Like, otherwise, in prokaryotes, what happened? This DNA will be arranged haphazardly, but when the histones are present, it will keep the DNA in a particular place. Yeah, it fits inside. So that is the use of histones. When we talk about cell division, means reproduction, in prokaryotes it is binary fission, and eukaryotes it can be mitosis. Sexual reproduction is the transfer of DNA fragments in prokaryotes, and in eukaryotes it involves meiosis. Okay, so please take some time, and I will anyway share these slides, but take some time to understand this prokaryotes and eukaryotes difference, so that we can move forward to next question. You can click picture also for this for now, so that this will help us solving in other problems in the upcoming slides. I'll give you a couple of minutes to note down this. And this is the difference between the prokaryotes and eukaryote cell. Okay, so you can see the difference. You can click the picture of this slide, so you can see in eukaryotes, when proper cell wall is there, it's simple. Okay, so coming to next question, the three domain classification was described by So the correct answer is option B, as all of you were right. Okay, I think there was a question. What is cytoplasmic streaming? So it's, I think it's the movement of the cell cytoplasm. So actually it helps to transfer the nutrients and the genetic material. Okay, so all this mm, uh, basically it's transfer of nutrients from one cell to another and from genetic material to the offspring. So this happens. So coming to next question, the organisms that can survive in high pressure conditions are called, they can survive in high pressure conditions. Barophiles, extremophiles, psychrophiles, or both A and B. Think more, think a little bit more. Barophiles is correct, but there is something else to it. Yes, option D is correct because barophiles, obviously, baro means pressure. Okay, so they will survive in high pressure. But when we say extremophiles, extremophiles means extremophiles contain all of those microorganisms that can survive in extreme condition. It can be thermophiles, it can be barophiles, it can be halophiles. So that is why both option A and B was correct. So that is option D is the correct one. Okay. So the next question is, MCR-A gene is linked for methane production in methanogenesis. This option is true or false? Think. So the answer is correct. So, as I have earlier asked why microscopy is important in microbiology, so here is the time for another descriptive question, is why three domain classification is important? Why not just two domain that we have seen till now, eukaryotes and prokaryotes? Why there was third domain introduced? Is there anyone who can answer? You can type in the chat box also. I'll give you 1 or 2 minutes to think. You can take some time to think. So we were aware that there are already eukaryotes, prokaryotes, and we have been studying them till now. This three domain classification was discovered very earlier, but we are studying only those two mostly, most of the time, if we are not going too deep. Okay, so why this three domain classification came? What was required? Yes. Yes, Sri Savitri, you have answered it very correctly. There are few microorganisms that were neither prokaryotes, neither eukaryotes. So we kept them in the third category. So that was archaea. And they were really strong, really strong. And archaea were one of the few microbes that were found when the earth was kind of after the Big Bang theory. After the Big Bang, when the origin of life, those were one of the few microbes that were the part of origin of life on earth, that are believed to be the part of origin of life of the earth, on the earth. No one is sure that that is there, no one has seen it, right? But those are believed to be the part of when the new earth was, new earth means the our earth only, it was found. So this is the three domain classification. One is bacteria, archaea, and eukarya. Okay, So they have a common ancestor. It is believed that they have a common ancestor. So initially it was just bacteria and eukarya. In eukarya it came fungi, animal, all those with proper membranes. And in bacteria it came cyanobacteria and other bacteria, purple bacteria, gram positives, green non-sulfur bacteria, all those things. But there was few bacteria which were in between the two. They had the property of bacteria and eukarya. So it was said that archaea have more suitable membrane chemistry than bacteria. Okay, their membrane chemistry was better than bacteria. Bacteria, bacteria and eukarya have, which may make them better able to survive in extreme environment. Okay, so that is why they have, because they have good membrane chemistry compared to both the archaea and bacteria, they can survive in extreme environment. If we talk about bacteria, if we know that the water is contaminated, if we say water is contaminated, we boil it, we keep on boiling in it 5 minutes, 10 minutes, 15 minutes, the bacteria will die, right? Same way, if we by mistake if we put our finger in hot boiling water, we will be burned, and that will not be good for our for us. If we um, if there is some condition that we had to survive in more than 50-60 degrees Celsius, it won't be good for us. Okay, but these archaea, they were able to survive in very high temperature. Okay, high temperature, high salinity condition, kind of extremophiles they were. So they have more stable membrane chemistry than bacteria and eukaryotes. And this is the basic difference between the archaea, eubacteria and eukaryotes. Okay, eubacteria is prokaryotes only. So cell type for archaea is prokaryotic cell, and for eubacteria obviously prokaryotic, and for eukaryotes it was eukaryotic. Chromosomes for archaea and eubacteria, they were circular, and for eukaryotes it was linear chromosomes. Hmm? Cell membrane lipids for archaea, it was of glycerol and ether lipids, and for other two it was ester lipids, ester bonds were there. Ribosomes, 670S ribosomes, but small subunits is more similar to eukaryotic ribosomes. Archaea was kind of similar to eukaryotes in terms of ribosomes. Okay, in eukaryotes there were 80 ribosomes and 70 ribosomes in mitochondria. Cell wall in archaea was always present without peptidoglycan. There was no peptidoglycan in archaea. In eubacteria, when the cell wall is there, it is with peptidoglycan. And for eukaryotes, sometimes only cell wall will be there, but without peptidoglycan. And histones, in archaea and eukaryotes, yes, they are there. It means their DNA material is pitidable. For eubacteria, it's haphazard. No. So introns in archaea sometimes it's there, eukaryotes it's rare, and eubacteria it's rare, and eukaryotes it is there. So what is introns? Is there anyone who can write in the chat box? Yes, it's non-coding genes. So when this DNA material is translated into protein, so there are some non-encoding genes that are removed before protein translation. Yes, portion that not code for anything. Not anything is protein only. Mostly it's protein. So portion that not code for protein is called introns. So in eukaryotes and archaea, it is there. And for eubacteria, it's not there. So The next question is related to microscopy only. The range of electron microscope is 100 micrometer to 1 nanometer. Is that true or false? So the answer is true. All of you are right. The answer is true. The range of electron microscope is 100 micrometer to 1 nanometer. It should have been written in reversed way, 1 nanometer to 100 micrometer. It should go from lower to higher, right? So it should have been reversed. The next question is choose the right option. Here are in the left side of the panel there are different kinds of microscopic techniques, and the right side, their what do we say? Their properties. So please match this. Take some time, take some 5 minutes, match this properly, then write the answer in the chat box. Okay, I have so many responses now, so we'll see the answer. So answer is A, correct. Differential interference contrast is based on refractive index. Why? Because when when it is passed through a sample, there will be differential. Differential means different. So to the top surface, middle surface, bottom surface, there will be little difference. So that is why it is refractive. It is based on refractive index. Light reflectance will be different. Like that, atomic force is used for the topography mapping. A confocal scanning laser is used for sample containing fluorophores. And D is electron microscope. It is used for, as a scanning, that is called uh, SEM analysis. And when it is used for transmission, it is called TEM analysis, transmission electron microscopy. Okay, so these are the pictures which shows different kinds of microscopy and the bacteria or the organisms that we can see. First is TEM analysis, transmission electron microscopy. Here it the capture the light is passed through different layers, so that is why we can see insides also. Inside thing also we can see very clearly. When we say scanning electron microscopy, it is at the outer layer. So how the outer layer is developed, that we can see in scanning electron microscopy. Other thing is dark field microscopy. The dark field microscopy, the background area is really dark as compared to the object that we want to see. And differences in bright field microscopy. Okay, it is just the opposite of dark field microscopy. Other thing is fluorescence microscopy. So in fluorescence microscopy we can see different colors. Next is phase contrast microscopy. So this is also kind of transmission electron microscopy, but here different objects, if they are kept one over another, all those things can be clearly seen. Inside of the object will not be seen, but the object behind the certain object can be seen. So if we want to see inside, we can go with transmission electron. If we want to see the colors and all, we can go with fluorescence microscopy. See, for normal practices, we use dark field, bright field, fluorescence, phase, all four for normal practices. Okay, if uh, I am sitting in my lab only, if possible, I will show you at the end of class. I will show you the microscope that we have. It is a digital microscope. Okay, here we have these four kind of microscopy: dark field, bright field, fluorescence, and phase contrast. So for normal work, we can use this. Okay, but if we want to do anything very specific, very research based, very novel work, then we go for SEM or TEM. So for us also, when we want to do this normal work, we use our microscope. But when we want to do this SEM or TEM analysis, it takes time, and we have to pay money also to different services, because these are kind of costly. So to maintain these experiments, they charge some money from us. Okay. So for research based work, SEM or TEM is used. For normal, these four are there. And these days microscopes are there in which all these four things are in one system only. In very one small system, all these four things are there. So this is also a slide I have prepared for you to know the difference between first light microscope. So the one which we have in our lab is the light microscope. Here we have bright field, so commonly used in wide variety of laboratory application. It is a standard microscope. The image is against the bright background. So the image will be red, blue, green, dark color, but the background will be bright. Next is dark field. So here what happens? The background is dark and the image can be of multiple colors. You can see the diagram, right? Next is phase contrast. So it uses refraction and interference caused by structures in the specimen to create high contrast. So what happens? If there are any objects, the light will be reflected in different intensity from the surface, from the sides and everything. So that is why when the light reflectance is different, we can see the structure of the object, the outside structure, not inside. So here we use phase contrast. Okay. The next is differential interference contrast. It uses interference pattern to enhance contrast between different features of specimen to produce high contrast images of living organisms with three-dimensional appearance. Okay, if we want to see something in three-dimensional and that too living, see what happens when we do gram staining and all, right? It kills the microorganisms. But if we want to see live organisms, we can, this gram staining and all can be used in bright field, dark field, phase also. But if we want to see live organisms, we want to see live moving organisms, we use differential interference contrast. It does not require staining and it won't kill also. Other thing is fluorescence. Okay, so fluorescence stains. What happened? Here fluorescent dye is used to stain the microorganisms. So it produces image which can be first, see, when we stain, all the things will die. When we do not stain, we don't know if something is living or something is dead. But when we do fluorescence, na, the living organisms, we are putting same dye. Let's see, we are putting green color only, but the living organisms will take different type of green and the dead organisms will take something like purple or other color. So it is very easier to differentiate if the object is living or dead when we use fluorescence, because it is the property of their cell membrane, cell wall, so that if it is alive, it will take different colors. If it is dead, it will take different colors. The next is confocal. It uses laser to scan multiple Z planes successively. Okay, so what is Z plane? You know, right? X axis, Y axis. So that thing is Z axis. which it talks about the depth of the organisms. If the organisms, how thick the organisms is. So for using that, we use confocal. Next is two photon. It is a scanning technique in which long wavelength of light is used. So there are some times when we take microorganisms, they will be single or two, three, but sometimes it is in the form of biofilm. So where you can see the biofilm, okay, I'll give you an example. If you might have been to villages and all, if we take water from hand pump, okay, and we leave that water into the bucket, and if we leave it for 2 days, 3 days, 4 days, 1 week, when we will throw the water, and if we touch the surface of the bucket inside, it will be sticky, slippery. So that slippery, sticky surface is nothing but bacteria in the form of biofilm. Okay. So normally bacteria is there everywhere, air, water, everywhere, but it won't feel sticky, right? But when it becomes the form of biofilm, it becomes sticky because it is now very thick. So for looking into those things, we use two photon microscopy. The next part is difference between TEM and SEM. TEM is a transmission electron microscopy and SEM is scanning electron microscopy. Okay, in SEM, the electron beam scans over the surface of samples. So we can see what kind of surface it is. Is it oval, round? What is the structure and everything? Next is TEM analysis. So here electron beam passes through the thin sample. It passes through the sample. If the sample is thin and if the sample is thick, what we have to use? Two photon. Next is in TEM, it is specially prepared with thin samples are supported on TEM grids. Okay, and in SEM, in SEM, sample can be of any thickness because it can be thick. Okay, we are using uh, we are just checking the outside portion, right? So that is why it can be of any thickness. I have a question, ma'am. In electron microscope, we use electron beam. In two photon. Now, see what is meaning of photon? Photon means light. So we are using light, but not in visible range. Not in visible range, but in other ranges. Infrared, like infrared range we are using. Okay. The wavelength will be more than 700 nanometers. So we are using in that range. Maybe we will use different kind of wavelength. Infrared, near infrared, far infrared. Okay, so that is how this will give contrast between two type of objects. Okay, in TEM analysis, specimen stage halfway down the column. So what happens? There will be a column in which TEM or SEM analysis is done. A column just kept under the electron beam object. So specimen is at the halfway so that the light can go from both the direction and we can see through it. And in SEM it is at the bottom so that electron beam can go from just one direction. And there are how the screen is there, it is fluorescent screen for TEM, and for SEM it is a simple TV monitor. In TEM we get two-dimensional projection of sample, and in SEM we get image is of the surface of sample because we are talking about just the surface. In TEM we are going inside, in SEM we are just being on the top. So you can see in this picture, right? In transmission TEM we can see the inside objects, but in SEM only the outside is visible. So the concept of microscope is finished. So the next question is, dash has an additional layer of periplasmic proteins than the. Please write the answer of this question. Take some time to think. I will give a couple of minutes. Okay, I can see 50-50 chances of the answer. Okay, now 3 is okay, 50-50 again. So we'll see the solution now. So solution is B. Gram negative bacteria has an additional layer of periplasmic proteins than the gram positive bacteria. So to explain this, we will see further slides. We will see proper explanation. What is periplasmic protein and all. Okay, so for now, this is the solution. So first of all, what is gram staining? What is gram? Okay, so Gram is the name of the person, the physician Hans Christian Gram, who developed this gram staining in 1884. He found that there are two types of bacteria. One is gram positive, another is gram negative. It depends upon what? Periplasmic proteins. Okay. So that is one thing. This is about history. This is what he found. So what is gram staining? So gram staining is a procedure in which we take bacteria. So there is a kit available in labs that is called gram staining kits. So all these four things, crystal violet, iodine, alcohol, safranin, all these things will be present inside the kit. You just have to bring your bacteria. You have to prepare a slide. You need a cover slip. These three things you have to bring. Okay. So in this glass slide is there. I hope you have seen in your earlier, or you might have seen it now. So in the glass slide, we first put some few drops of bacteria. Okay. Then first process is the application of crystal violet, that is purple dye. So we first put some crystal violet, then we heat it a bit. We heat the slide a bit. So all the things will be evaporated and the glass slide will be dry. Okay, so that is first step. The second step is application of iodine. Okay, here iodine is mordant. What this mordant will do? It will just help crystal violet to stick to the wall. Okay, iodine is doing nothing. Iodine is not giving any color. It is just helping the crystal violet to stick to the walls. So these are two steps. After that, we will put alcohol. So when we will put alcohol, what happened? Some of the cells will decolorize. The color will go away in some of the cells. Even after putting mordant, in some of the cells, color will go away when we put alcohol. And the next step is safranin. So when we will put safranin, all those bacteria for which color has gone away, it will take color of the safranin. Okay, so this is the process of gram staining. So please note this down, understand this, so that we can move forward to next slide to see what else can happen. Take some time to comprehend this slide. First step is crystal violet, second is mordant, third is decolorization. So So, okay, so not all of the bacteria will lose their color when we apply alcohol. Some bacteria will lose. Then in the next stage, when we add safranin, these bacteria which has lost their color, they will take up the stain. So some of them will have purple color and other will have the safranin color. Take some time to note down. Okay, so the gram positive bacteria are those bacteria which were taking the first color, the purple dye. And the negative bacteria are those bacteria which were decolorized by using alcohols. Then they took the color of this red, safranin. So, what are gram positive bacteria? They have thick peptidoglycan layer in their cell wall. So, because they have thick layer, this alcohol was not able to remove the color. However, for the gram negative bacteria, because they have thin peptidoglycan layer, this alcohol was able to remove the color. So gram positive bacteria, they retain the purple color of the crystal violet stain, and the negative bacteria, they did not retain the color of the purple crystal violet stain. For the gram positive bacteria, they have porins in their outer membrane. Okay. And for the gram negative bacteria, they do not have porins in the outer membrane. So what is this porins? Is there anyone who can write in the chat box? From the name itself, it is kind of available. What is the name? Yes, correct, pore. So they are protein molecules that form channel or pore in the cell membrane. So gram positive membrane, they have porins for transport, and gram negative membrane, they don't have porins. Okay. Gram positive bacteria, they produces endotoxins, and the gram negative bacteria, they produce exotoxin. Gram positive bacteria are more sensitive to lysozyme, and gram negative bacteria are more resistant to lysozyme. What is lysozyme? What is lysozyme? Yeah. So they are the proteins that protects against other organisms. Okay. Hydrolytic enzymes present in lysosome. Yes, correct. So they protect the cell wall from the external effect. Lyso means to break. The the organisms which have these lysozymes, they tend to rupture the other organisms on which it is attacking. Okay, lyso means break. So always go towards the root word. Okay. So gram positive bacteria can be attacked by some lysozymes, but gram negative bacteria cannot be attacked. So this is the principle of gram staining. So when the bacteria is stained with primary stain crystal violet and fixed by mordant, some of the bacteria are able to retain the primary stain. Okay, so this is the first step. After that we are applying alcohol. So some of the bacteria are decolorized by alcohol. The cell wall of gram positive bacteria have thick layer of protein sugar complexes such as peptidoglycan, and lipid content is low. In gram positive bacteria, lipid content is low. Okay, so decolorizing the cell causes this thin wall to dehydrate and shrink, which closes the pore in the cell walls and prevents the state from exiting the cell. So we have seen that, like in gram positive bacteria, they have porins. So what happens when we decolorize them using alcohol? This bacteria will dehydrate. When something dehydrates, it shrinks. Okay, for example, if you have a resin or any dry fruit, when after putting in water you keep it in sunlight, it will become small. So what happens? It becomes shrink. So the pores will close. When the pores are closed, then this stain, this purple stain won't come out. Okay, so because they have pores. So the ethanol cannot remove the crystal violet iodine complex. What is ethanol? That is the alcohol. Okay, so what happens in gram negative bacteria? Cell wall take iodine complex. Okay, it is also colored blue, but due to thin layer of peptidoglycan and thick outer layer which is formed of lipids, crystal violet iodine complex get washed off. Because the layer is thin, this iodine complex get washed off when we dip it in alcohol. So when they are exposed to alcohol, decolorizer dissolves the lipid in the cell wall, which allows the crystal violet iodine complex to leach out of cell. So then when they are again stained against safranin, they take the stain and appears red in color. Okay, so there is lipid in gram negative which is dissolved in alcohol. So that is why the color which is present inside the gram negative, it comes out. You can see in this picture, right? Gram negative, these are big rod shaped, it comes out. So that was all for the gram staining. Okay, you have seen the answer, but no problem, I will still repeat the question. Which of the following is metabolically advantageous for a cell? Please try to find out answer of this question. High surface area, less volume, none, or both A and B. So the answer is correct, both A and B. So there is a concept called surface to volume ratio. So if the surface to volume ratio means, if this surface to volume ratio is higher, then it will be helpful for the transport and all. Okay, so that is why the bacteria which are of circular size, they have high surface area and lesser volume, they are resistant. So most of the high quality bacteria, high resistance bacteria, they have high surface area and less volume because of this circular shape. So the next question is, this question is from the central dogma part. So the question is, start codon binds to the starting sequence of DNA in the protein synthesis. try to find out answer of this question. Read the question very carefully, then answer this question. Those who are answering false, please let me know what will be the correct statement. What word do we have to change? Correct. 100% correct. So the answer is false. Why? Start codon binds to the starting sequence of mRNA in the protein synthesis. Yes, yeah, tRNA. In the protein synthesis, not the DNA. So obviously, start codons are the starting sequence of protein synthesis, but not DNA. They are bound to RNA. So that is why this statement is false. Very good. Coming to next question. Small segments of DNA that can move from one region of DNA molecule to another region is plasmid, pili, transmopson, chromosome. And what is another name of the answer to this question? Very common name. Correct. So the answer is transposon and the other name is jumping gene. So it is very clear from this name itself that it moves from one region to another region. It jumps from one region to another region. So that is why this name, jumping gene. Moving towards next question. Identify the wrong pair. A, B, C, D. Which one is the wrong pair? We know that when these base pairs combine in DNA, they combine in a particular pattern. So which of them is showing some wrong pattern? Think more. Try to find out answer of this question. Take some time and think about this question. Think about all the base pairs. Yes. The wrong pair is option C, adenine, thymine and purine. So this is the wrong pair. We will see how it is wrong pair in the upcoming slides. So after this we will see that. So the next question is identify the wrong statements. Nucleoside attached to phosphate group is nucleotide. Two strands of DNA are parallel and connected by hydrogen bonds. Protein is not a polynucleotide. tRNA helps in the transcription process. Please try to find out answer of this question. Okay, see the solution now. So answer is B and D. So okay, option D is fine. Why option B is incorrect? What will be the correct word? about the option B? Correct, 100%. Two strands of DNA are anti-parallel and they are connected by hydrogen bonds. So in this slides, we can see first is in the left part of the slide, DNA polymerase. So there will be first replications, means one DNA will form multiple DNA of the same type. Then when this DNA is converted to RNA, this process is called transcription. So DNA is double helical and RNA is single strand. When this RNA is converted to protein is called translation. So replication, transcription and translation. So these are the process. Coming to the right side first, so these are the base pairs in which this base pairs can be bound. So we can see some of them have triple bonds and some of them have double bonds. So these are the base pairs which can be combined. Okay, G always combines with C. You can see in this picture also they have tried to replicate it very nicely. G is going inside C, so it's like fitting in the form of puzzle. Okay, AT, CG. So it's like a perfect fitting puzzle. So these are the base pairs. So here this option C was not correct, right? Because this thymine purine was not fitting in the form of a good puzzle. This is just a analogy that I am giving you. Now anti-parallel DNA strands. Why? Here 5' is at the top and the other side 3' is at the top. So mathematically, even if they are parallel, they are in front of each other. So this prime, 5', 2', these things, because of this, it is anti-parallel, even though they are in the same direction. And we can see hydrogen bonds, other things, okay? Double bond, triple bond, here also in the next picture also we can see. So this we should remember that it is easier to break double bond but not easier to break triple bond. Okay? So all those things we need to keep in mind. So that's all for today. So I will give you my website in case you guys are having any doubt. You can just let me know. You can type your question in this website and I will definitely answer. I will answer you via email. I'll get your email with this, because while typing question you guys have to put your email also, and I will get the question and I will surely reply, and if required we will learn about that particular thing, whatever your doubts are, in the next session. Okay, so that's all for today. I'll still wait for a few more minutes in case you guys are having any doubts, then we will stop the session. I'll stop presenting and I'll wait for some time. Yeah, we will upload the slides, then these slides will be given to you by the NPTEL. Okay, maybe when I'll upload today, you will get it by tomorrow or day after. Okay. Thank you, everyone. Thank you for being here and being such a nice and interactive audience. I hope in the next session also I will see all of you, and I'll try to improve the slides, I'll try to improve the information and everything, and I hope you guys can get better out of it. Okay, so I'll close the session now. Thank you, everyone. Bye bye. Take care. Good night.